Computer. Okay, so today we're going to continue our discussion of MRI. Before we begin, are there any questions about course logistics projects? I think there's still maybe a bunch of groups who need to submit revisions on your proposal. So if you have questions, just come talk to me or email me, or if you're on Zoom, you can ask me on Zoom. Um, Otherwise, I think everyone looks pretty good. Just try to make sure you make those revisions as soon as possible. Okay. Um, so yeah, so today we're gonna sort of talk a little bit. We're gonna finish talking about image contrast, and then we'll get into functional MRI. And these are basically making use of relaxation. Okay, let me sort of, whoops, what's going on here? Scar, let's try this again. Hmm. All right, well, we'll keep the color way it is. All right, so these are going to be, we're making use of relaxation to create contrast in our images. And then if we have time, we will probably start talking about diffusion, which is going to be looking at the motion of water. Okay. So it turns out MR is really, so in this class, we're only going to be able to talk about a few contrasts that you can do with MR. It turns out MR can do like hundreds, if not thousands of different contrasts. And so there's a lot of information because there's the waters moving, there's the chemical environment, there's relaxation. So there's a lot of stuff going on at the same time. And so in this class, we'll just introduce you to a few of those contrasts. And some of the projects might look at some other contrasts as well. Just a word about the pre-lecture quiz. I think everyone did pretty well on most of the questions. I think there was just the one question on grappa where I think everyone got the sense that if you have a coil profile that is sort of shallow or doesn't change very much, then it's going to have, like some Fourier transform like that, right? And then if it's broader, then the Fourier transform, uh, it's even narrower, right? Okay. And so the broader transform is better for grappa, right? Um, but the trick, sort of maybe the trick of that question was that the question only asked you about coil one. So all you had to do was look at the coil one profile. You didn't have to like look at coil two profiles. So if you did that, then you would definitely pick the one that let's see, that sort of had the broader profile in K space. Okay, so this is the Fourier transform. All right, so any questions about that one? All right. Okay, so let's move on. So as we talked about last time, first we're going to look at relaxation rates. And so we can have the ones we talked about are T1, T2, and T2 star. In addition, we have proton density. Okay. And so the way we're going to talk about adjusting contrast is by adjusting the echo time and the repetition time. All right. So we talked about how a saturation recovery sequence looks like this, where we're going to assume that after we apply the 90-degree pulse, then the magnetization is at m sub 0, and then at this point, right after we apply the 90-degree pulse, there's 0. So right at this point here, then mz equals 0, and mxy equals m naught. Okay, so basically that's just saying all the magnetization that we have available is tipped over. Okay, so that's the first pulse we apply because we assume there's full magnetization. But what happens is that m sub z starts at 0 and it's only going to recover. So let's say m sub naught is here, right? This magnetization, if we wait out TR, it may not fully recover. Okay, it's not guaranteed to recover if we don't allow enough time. And the shorter we make the TR, then there's less time for it to recover. So the magnetization we have at this guy here, at this point here, now this m sub z is going to equal this thing. Okay, because that's the sort of the m sub z right before the 90 equals that. And so right after the 90-degree pulse, then the mxy will also equal that. Okay, so the m sub z recovers to that 1 minus e to the minus tr over t1, and then that magnetization is what we flip over. And so that's where this term comes in. And then once that magnetization is flipped over, then it decays with the T2 star decay. All right. And then if we apply a 180-degree refocusing pulse, then what that does is it just changes that T2 star into a T2. Okay. The longitudinal relaxation part is still the same. Okay, so for T1 weighted scans, the idea is we want to make TE very short compared to either T2 or T2 star, such that that EXP minus TE over, say, T2 is approximately equal to 1. All right, because if we make, in the limit, if we make T equal to 0, then E to the 0 just equals 1. And so you sort of have something here that only depends on the proton density and T1 and TR. Okay. So we want to talk about how do we, if we have two tissues with different T1s, how do we maximize the contrast between those two tissues? Okay, this is just reminding you what we mean by T1 relaxation. So basically it's the recovery of the M sub Z. And so in this case, M sub Z is recovered to this point. And so when we tip it over, we have this full M sub Z, the amount of M sub Z to work with. If we have a very long T1 in this case, it takes longer to recover. So at the time we're ready to tip it, when we tip it, there's much less to tip over. Okay, so that's basically telling us how much signal we have to work with. Yeah, question. That's a great question. So if we wait for it to recover, there's two things. One is if everything recovers, then there's no contrast, right? Because all the tissues, whether depend, regardless of their T1, are all back at full magnetization. So then there's no T1 contrast. The other reason, which we'll see later, is remember, if we're acquiring one line every TR, then the longer we wait, then it takes the scan, makes the scan much longer. And in fact, we'll see later in this lecture where there's this type of scans where you just go as fast as possible. Okay. So just to give you a sense of what T1 contrast looks like, here we plotted the T1 relaxation of, this is an 800 millisecond T1, right? And this one is a T1 of 1200. And as you expect, the one with the longer T1 takes longer to recover. So for contrast, we can look at the difference between those two signals and see where it's maximized, right? And we sort of look as a function of my TR, where is that difference maximized? So this is the difference. And we see it's maximized around here, okay? So in this case, it's sort of the average of the two T1s because the average of these is like 1,000 milliseconds, right? Okay. So assuming the proton densities are the same, then a good rule of thumb is you just sort of take the average of those two T1s. And so that's, so you can sort of see here to answer your question, if I took a very long TR at 5,000, they both relaxed, we've both recovered quite a bit. And so you can sort of, the difference is very small. Okay, so that would not be good for T1 contrast. So here's an example. I mean, T1 contrast is used in a lot, but I'll just give you some examples here. In fact, what we talked about is that fat has really short T1s, right? Because the hydrogen protons that are sort of attached to the fat chains, the hydrocarbon chains have the tumbling rate that's sort of close to the Larmor frequency, and so they can promote sort of the relaxation back to equilibrium. And so in this case, for example, here's a head that is showing a very bright spot in the middle of that person's brain. So that's not normal, okay? So essentially that just means that this means that in this part of the brain, there's something with a very short T1. Okay, much shorter than the surrounding brain. So the brain area, these T1s are around, maybe 1,000 milliseconds. Okay. So that means that that bright area in the middle must have a T1 shorter than 1,000. It's much brighter than the surrounding. And in fact, it's probably got a T1 of about 300 or so. And that's basically a case where that part of your brain is what's called the corpus callosum. It's like the highway of fibers that connects your left and right hemispheres. Okay. And so in this person, for whatever reason, they have what's called lipoma of the corpus callosum. There's just a lot of fat in their corpus callosum. So that's sort of the diagnosis there that that's an abnormal amount of fat in their corpus callosum. Okay, so that's T1 weighted scans. For T2 weighted scans, we want to make the TR very long, right? Because we said if we make TR really long, we're going to get rid of T1 contrast. And then therefore, that 1 minus EXP minus TR over T1 term goes to 0, or it goes to 1, sorry, because this term goes to 0, right? So then we're just left with the proton density and something that depends on, say, T2, okay? And so once again, we can sort of look at two species, one with a T2 of 80, the other with a T2 of 120. And the thing to remember is the longer T2 species decays much more slowly. All right, is what we see here. And once again, we can look at the difference, and you can sort of the difference is maximized here at around 100 milliseconds, which is once again is like the average of these two. Okay. And so notice here we're plotting against echo time. All right. So echo time is after we tip the magnetization, at what point do we acquire the center of K space? Okay. And so you can sort of see here, we don't really want to make the echo time too long, right? Because all the signal will die away, right? And so later on in the class, we'll talk about SNR, but essentially there's no signal there. And in fact, if you take the difference, there's also very little contrast as well. Yep. The sample image of which produce the best contrast. Yeah, exactly. Yeah. So basically, after you tip the magnetization, you're going through K space, right? And so TE is at what point you go through the center of K space. Okay, that's called the echo time. Okay, so echo time is we'll define TE is defined as when you go through the center of K space. In specific, where Kx equals 0 for spin warp. All right. Because remember, the center of K space tells you sort of the overall brightness of your signal, of your image. So that gives you a good sense of where you're at. Okay, so here's another case where let's look at the normal case on the left. So you notice that there's some really bright areas. Okay, so looking at this, those must have really long T2s, right? Because they have survived. Okay, so in fact, what you're seeing is the areas that look really bright on that left side are the areas with a lot of fluid. Okay, so in fact, in your brain, you have what are called ventricles, which have cerebral spinal fluid in them. And so these are basically the ventricles, right? And they have what's called CSF or cerebral spinal fluid. So this sort of bathes your brain. And in fact, just over the last 5 or 10 years, it's become a really active area of research where it's thought that every night your brain sort of flushes itself, right? It sort of tries to get rid of toxins. And one of the hypotheses out there is that if that's impaired, for example, older people don't tend to sleep as well as younger people. And so there may not be this as a sort of effective flushing of their brain that occurs at night. And so toxins might build up. And that might be one of the things that leads to, typically as people get older, they get dementia or Alzheimer's or other diseases. And so that's one of the things. Now, the question is, what comes first? Is there impaired flow first and then you get worse sleep? Still not known. Or do you get the toxins in there first and then that makes it so that you can't flush as well? So I think this is an area called, if you're looking for what it's called in the literature, it's called the glymphatic system. This is a very hot area of research right now. Okay. Now, on the right, we're showing someone that's just has what's called an arachnoid cyst. Okay. And basically, all of this is filled with fluid. Okay. This is all fluid based. Okay. So the idea is the takeaway home from this picture is that T2 is really good for looking at fluid. Okay. And because remember, water has like a T1 of like 3,000 or 4,000, whereas everything else is sort of a T2, a really long T2 on the order of say like 2,000 milliseconds or so. whereas everything else is going to have a T2 of like less than 100 milliseconds in general, right? And lastly, we can make TR really long, and we can also make TE really short. So we're going to end up with something that just is giving you something that's weighted by the density of protons. Okay, so this is what's called proton density weighted scans. So those are the three major types of scans, and typically most clinical protocols will have one of each, just because they're all quite valuable. All right. This is a case where, for example, this area here that they're highlighting here is showing more density of protons in this subject with multiple sclerosis. And the idea is, for whatever reason, there's just the white matter, which we'll get into later, which is like the wiring of their brain. For whatever reason, there's more proton density in this person's white matter. Okay, so let's go back to this picture, which we showed earlier, and now we can sort of dive into and sort of try to understand this picture a little better. So what I'm showing you here is exactly the same brain, and all I've done is I've changed the TR and the TE to try to emphasize different tissues. Okay, so let's look at the T1 weighted scan first, right? So here, remember, things that are dark must have a very long T1. Okay. And in fact, what we're seeing here is like the fluid, right? The fluid areas, what we talked about, have are very dark. And so does that make sense? Do they have a longer T1? If we look at the T1 of CSF, yeah, it's got a really long T1 compared to the gray and the white matter. Okay. Similarly, let's look at the density weighted one. So this is density weighted here in the middle. This tells you this should be weighted by the brightest tissues should have the most density of protons. Okay. And so you sort of see here, it includes the fluid areas, right? It's because fluid has a lot of density of protons. It's lower here in the white matter here because white matter is made up of a lot of myelin and other things, and it's like the wiring of a brain. It just tends to have less protons per unit volume. And then here you're seeing some sort of mid-level brightness in the gray matter. That's where all your neurons are. That's where all the computation occurs. Okay. So if we look at the density of protons, we see, yeah, it is highest in the fluid, and then it's slightly lower in the gray matter, and then the lowest in the white matter. So that also makes sense looking at that image. All right. Now let's look at the T2 weighted image, right? Once again, what really stands out there is the ventricles, the fluid areas appear really bright. And that makes sense because their T2 is really bright. Okay. And then what's the darkest? The darkest is the white matter. This is the darkest area, and it's got the shortest T2. And then gray matter is somewhere in between, right? And you're also seeing bright areas here, right? Like little stripes of bright areas. That's because there's also fluid. Like your brain is like folded all up and it's got all these wrinkles. And so there's fluid in those wrinkles too. So that's what you're seeing there. All right. And so the question is, what sort of TRs and TE should we use to achieve this T2 weighted image, T1 weighted image? So for T1 weighted, remember we want to say, we want to make TE as small as possible, right? Um, and then we want to make TR as an initial guess. It's probably going to be somewhere between these two T1s, right? Somewhere between that. And so maybe we'll just guess, maybe around 1,000 might be good. Okay. Now that'll somewhat depend on what the proton density is, right? And notice I've only looked at these two guys. I've sort of ignored this one here because this is always going to be really dark anyways. So if I'm trying to get the contrast between gray and white, then I sort of need to sort of look at those two tissues and maximize that contrast, right? Because if I maximize that contrast, the fluid will appear dark anyways. So I'm fine with that. Okay. Density weighted is really easy, right? I want to make TE as small as possible. And TR, we could set it to infinity, but in fact, we don't really need to set it to infinity. So we could just look at the longest T1, which is 4,000, and maybe set it to like 3 times that. So maybe I'll set it equal to 12,000. Okay. In fact, the scanner will limit you to that. The scanners, if you look at any modern, well, most MRI scanners will actually have a maximum TR. Okay. Simply because, they have digital counters and stuff, and they don't want to they don't want to have a TR of an hour because no one's going to use it. So why set up the circuitry to do that, right? So there's going to be some countdown timer that they're going to use. And then for the T2 weighted image, um, obviously we want TR to be pretty long again. And then for the TE, once again, we want to sort of look at the contrast between the two tissues that are similar, right? Because no matter what, the CSF is going to appear really bright, right? So in this case, maybe I want to make TE roughly maybe like 95 or so, sort of the average of those two, okay? But once again, to sort of actually look at the difference, I would look at the gray and the white matter um, densities as well, okay? So this is an example. I think there's going to be one. We'll issue one last. It's going to be totally optional extra credit if you want to do it. You can. If people want to sort of get extra credit points, there's going to be a very small problem where it'll work you through how contrast works in a MATLAB problem, and then ask you to look at some equations if you want to. So what you're going to be given is a segmentation map like this, where basically we've already segmented into the blue is showing the CSF, the green is showing the white matter, and the yellow is showing the gray matter. Okay. And you're asked to sort of come up with TRs and TEs that give you either proton density weighted or a T2 weighted or a T1 weighted. And you can sort of use these rules of thumb to sort of guess what works. And then there's also in the code a program that sort of picks the optimum TR, and you can sort of compare your answer versus the optimum TR and look at how that was derived if you want to. All right. So if for those of you who want to sort of understand this better, there is an opportunity for that, but it will be optional. Okay. Okay. Any questions before we move on? Okay. So the next thing we're going to talk about is the flash sequence. And this gets to the question that was asked earlier about, TR. Okay. So the idea is, yeah, even though, let's say in the previous example, we said, yeah, I want to pick the TR of 1,000, that has a cost to it because if I have to wait a second before every line of case space, um, like let's say I wanted to acquire 200 lines of case space, then I have to wait for like 200 seconds, right? So that's going to be like almost 4 minutes, right? So uh, in general, we want much faster scans. So one of the most common scans, if you have an MRI, they put you in the scanner, and there's going to be a few initial scans that are done, which are called localizer scans. And they're going to be very quick. There'll be a few seconds each. And those are basically acquiring images in all the three planes, axial, coronal, and sagittal. And they're just trying to make sure they know where your head is in the scanner. Okay. And then everything is based on what are called localizer images. So we have these localizer scans. Okay. So that allows a technician to know where your head is so that they can actually um, prescribe where to scan. Now, with AI, a lot of that's going to be done by AI. Certainly within the next 5 years, probably almost all done by AI, where the scanner will just scan you and do most of the acquisition without even the technician interfering very much. Okay. So here's the thing that remember before we had a 90-degree flip angle, now we're going to go theta. We're going to do a less of a flip, okay? And then we're going to make TR relatively short. We'll still have the TE. And it turns out if we do that, we end up with this equation here, which is the intensity that we had, which is just longitudinal relaxation, now is replaced by this more complicated expression, which has the sine theta and the cosine theta in there. And there's still the T1 dependence here, but you can sort of see it's sort of modified. And then it turns out that there is an optimal angle that will maximize a signal, and it's called the Ernst angle. Okay, named after Richard Ernst, who also won the Nobel Prize for his work in this area, but for something else, actually more for chemistry. So why would you do that? So the idea, the innovation behind this was that if I totally flip over my magnetization, I start off at zero and I got to wait a long time to rebuild it, right? Okay. But what if I only tip it a little bit, right? Like let's say I tip it 45 degrees. I still get like 0.707, like square root of 2 over 2 here, right? And I start off at 0.707 here. So therefore, I've got a lot of magnetization for the next round. Okay. It's similar to is everyone familiar with like a flush toilet, right? Okay. So if you have a toilet and the water is completely full and you do a full flush, all the water goes away, right? You get a really strong flush of your toilet, but now you have to wait maybe like a minute for it to fill back up, right? Whereas now you can sometimes get like low flush toilets where you do it and it only flushes a little bit, right? And so this flush is not as strong, but it's usually good enough. But the tank is only gone down a little bit. So now you can flush again very quickly. Okay. So that's the basic principle behind the flash sequence. You say, okay, I'm willing to give up a little bit of my signal, um, and in return, I can have a lot of longitudinal component left, and therefore I can make the TR much shorter. And so now instead of a 4-minute scan, maybe I can go down to like a 30-second scan or even a 10-second scan. Okay. Because you don't, especially for things where you don't really want to spend much time on them, like a localizer scan, you want to make that trade-off. All right. So here's an example here showing the flash signal intensity versus flip angle here. Okay. And for different TRs. So you can sort of see each one here has a maximum signal intensity. And so these would occur at the Ernst angle. Okay. So that would be at the Ernst angle. Okay. And you can sort of see here if and this is the signal intensity versus sort of for a T1 of 1,000 milliseconds, with respect, one would be full magnetization. So you can sort of see here, if I do a TR of 500 milliseconds, which is pretty short, I get half of my full signal intensity, which is pretty good. But notice if I go down to 20 mils, if I go down to 100 milliseconds, which is even shorter, I'm still left with enough, like a little more than zero point 0.2 of my magnetization. And I'm willing to go down to 30 milliseconds, I still have like 0.13 of magnetization to deal with. So that's the trade-off. There's less magnetization to deal with, but you've gone much faster. Okay. The other thing that is sort of you should know about is what's called inversion recovery, which is sort of an interesting way of doing things. So essentially, we've looked at this before. So this is the Spinnaker pulse sequence. Now, what I do is before I even acquire my data, do my 50, I apply what's called an inversion pulse. Okay. So basically, I take my magnetization, it's here, and I invert it, right? So the magnetization starts at a -1, and now it's going to recover. At some point, it's going to cross 0. All right. So I can take advantage of that because now it turns out that if I set up this time Ti between the time I invert and the time I do my next flip, if that inversion time is equal to this expression here, then that corresponds to I've inverted the spin and I acquire at this time, at this inversion time Ti, if it equals that equation, then my signal will be 0, right? Because there's no M sub z here. So there's when I do the flip magnetization, I'm flipping 0. So 0 just goes to 0, right? So that's shown here where, for example, and then because different tissues have different T1s, I can decide, do I want to make CSF 0? Do I want to make white matter 0? Or do I want to make fat 0? So it's a sort of an interesting way. You can actually erase parts of your object by just picking your inversion time correctly. All right. So you can sort of see here, all I'm doing here is I'm changing the inversion time from 180 to 400 to 650 to 2500 milliseconds. And you're sort of seeing different parts of the image are becoming dark, right? So in fact, here, in these early images here, let's see, where's CSF? Oh yeah. So let's just pick this one. So this one here, you're sort of seeing the CSF is very dark. Okay, the fluid at that point, I'm at this long inversion time, and so you see the fluid is nulled out. Okay. Here, I'm mostly nulling out the fat. Okay. Fat is approximately 0. And here, the white matter is approximately 0. And here I'm making the gray matter approximately zero, right? So this is an interesting thing because sometimes you want to, for example, null out different areas so you can sort of see the remaining tissue better. These are some other examples of that. And so one area where it comes out is, for example, when people are doing cardiac imaging, they really want to see the vessel wall, right? And they don't want to see all the blood swirling around. And so in this case, we would do this is sort of called a black blood sequence. So you basically time it so all the blood sort of disappears, and so you get a better visualization of your vessel wall. Okay. Okay, so that's all we're going to talk about for relaxation, and now we're going to switch gears and talk about fMRI. So before we move on, any questions about that? What we talked about? Okay, so pretty much the distinction between MRI and fMRI is before fMRI came about, MRI was mostly a study of like brain anatomy and some physiology. And functional MRI sort of was the first non-invasive way using MRI to really effectively study what the brain is doing. Okay. So functional MRI, what it looks like is we typically use a standard fMRI machine or MRI machine, but we'll just maybe add some additional features. Like there might be a projector that's shining in some stimulus. The subject might be just looking at something, maybe watching a movie, or perhaps playing a game, doing a gambling task. So there's lots of different things. They might be smelling different odors or even sipping different drinks, tasting different things, um, feeling pain, having acupuncture done on them. So anything you can sort of think of has probably been done in the fMRI system. Okay. So it's relatively new technology. So the first paper that sort of identified this thing was in 1992. And then the first human studies came out in 1993. Okay. So basically it's like 30 years old. All right. So here's the basic phenomena that they first saw, which was that here we're seeing sagittal images. So sagittal, so this is like a slice this way. And so you're seeing the brain from the side. Okay. And everyone, this is where your visual cortex back here. Okay. If you sort of put your hand right above where your hairs, with your neck and head interface, this nature has devoted a lot of real estate to seeing. Okay. So that's your visual cortex. All right. So that's what they're sort of homing in on the visual cortex here. And this was one of the first studies. So it was actually very simple. Basically, they just shined light or turned it off. Okay. And they're just sort of seeing, what part of the brain is responding and how does it respond. And so you can sort of see here, when the light is shining, the MR signal is going up. And when they turn off the light, it goes down and on and off again. Okay. The other thing they did was, and this is where now you can sort of get a sense, we can start to understand this at a slightly deeper level, is they changed the echo time. They acquired it two different echo times because they were trying to figure out what are they measuring, right? So notice when the echo time is 40 milliseconds, you see the signal, okay? And then when the echo time is 8 milliseconds, there might be something there, but it's a much weaker signal. So therefore, whatever they're measuring must have some echo time dependence. Okay. And remember, echo time dependence, things that are echo time dependent are related to T2 and T2 star because they're related to how fast that transverse magnetization is decaying. This is fairly old. This needs to be updated. But just showing that, fMRI is just, lots and lots of people doing fMRI. And typically, if you're looking at the news or anything, anytime there's like a brain image with like spots in it, is most likely fMRI. Okay. So one of the key trade-offs we make in fMRI is, we go from very high spatial resolution. So on the left here is something where we have fairly small voxels, say 1 millimeter by 1 millimeter cubed, And we but it maybe take us 6 minutes to acquire a nice looking image. All right. But in 6 minutes, your brain goes through a lot of things, right? I mean, I don't even remember what we were talking about 6 minutes ago, right? We were talking about some other like relaxation, or I don't even remember. So a lot happens in 6 minutes, right? So to actually study the brain, we probably need a finer temporal resolution. So, for example, this slice was acquired in 60 milliseconds. Okay. The trade-off is we went to a much fatter voxel. So this one here was say 45 millimeters cubed. So that would be like maybe a little less than 4 millimeters on a side, right? 4 millimeters cubed would be 64. So it's like 3.75 or something per side. Okay. And the way we do that um, is with some variant of what's called single shot equiplanar imaging. Okay, so this is very much related to the MATLAB grader problem you did for this current homework, which is basically instead of going line by line by line through K-space, which is going to take us time, we sort of zigzag through K-space all in one shot, if possible. Okay, and so we try to do this total time to go through, right? We try to make this on the order of, say, 50 milliseconds or less. Okay. Because remember, things are decaying. Our T2 stars are decaying. So we need to go through all of K-space relatively quickly to have enough signal to work with. So here this summarizes it. The MRI is sort of looking at anatomy. It's slow but high resolution. And in fMRI, we sort of acquire images while we're having the brain do something. And in this case, for example, we're showing a flickering checkerboard for 20 seconds, and then we're alternating that with just like a gray background. And if we look at some area in the brain that we can sort of see that the brain signal is going up and down. And then typically what we do then, the most basic analysis is we'll then just do a correlation. We'll ask ourselves what part of the brain has signals that seem to be going up and down the same way as our stimulus. Okay. And then we can come up with a correlation map. And, then there's a lot of statistics involved after that. Okay. And then so then you can plot like nice little images. You can make a 3D rendering of the brain. You can sort of plot out what areas had, activation that was correlated to your task. And once again, this is what we showed before. We saw that that is what's called the contrast we're seeing is what's called bold contrast, which stands for blood oxygenation level dependent contrast. And that sort of gives you a clue as to where we're going. Somehow we are sensitive to blood oxygenation. Okay. And so the clue is this echo time dependence. At 40 milliseconds, there's signal. At 8, not much signal. So what could be going on here? Well, remember we said that whenever there's decay, transverse decay, there's a T2 component, which is due to random motion, and there's a T2 prime component that's due to inhomogeneities. And the combination of those two is what gives you your T2 star. And the inverse of that is your R2 star, which is just the transverse rate of relaxation. So the rate of transverse relaxation. So basically, instead of writing things EXP minus T over T2 star, right? That's equivalent to writing it as EXP minus R2 star of T. Okay. So we're going to find it easier to sort of think about things in terms of rate of relaxation. Similar to like when you're dealing with chemical reactions, you look at a chemical rate. Okay. So let's remind ourselves of how signals decay in MR based on the phase of the spins. So remember, we looked at this example before. If I start off with all the spins in phase as I tip them, and then I expose them, I put them in some sort of inhomogeneous field, then each spin experiences slightly different magnetic field, and therefore it's going to either accumulate some phase or maybe lose some phase. Okay, it's going to clockwise or counterclockwise rotation. That dephasing leads to this sort of decrease in the MRI signal, right? Because now all the vectors don't line up. And if I increase the amount of inhomogeneity, then there's even more dephasing. And so the MR signal is going to go down. So what did we see in our experiment? We saw that when we shone light, the signal actually went up. Okay. So for example, let's say, so this must mean when we were in the dark phase, we were probably here. And then when we went to light phase, went to here. Okay. So if something like this is going on, it must be when the brain is active, somehow there's less inhomogeneity. Okay. So let's try, and so that was a big mystery. Like, why is the signal going up? Right. And so that's what people had to sort of first figure out. So we're going to dive into just a little bit of the physiology of the bold signal change so you understand what we're measuring here. Um, so remember, let's say in baseline, and so maybe like this is the dark phase, right? There's going to be some signal decay, and if we measure echo time, it's going to be the signal is going to be the echo time times my R2 star of baseline, and there's exponential decay based on that value. Okay, so that's shown here in green, right? And then we assume during an active state, let's call this like when we're looking at the light, that activation, the echo time we keep the same. Okay, that's the same. Now the R2 star is going to change. We're assuming there's a different relaxation rate during the active period when the brain's active. But since we know the signal goes up, that R2 star must be smaller so that the decay is slower. And so that must correspond to this red curve here. Okay. Typically, when we look at the bold signal, it's a percentage change. We're taking the difference in the signal, so this difference, delta bold, and we divide by sort of the baseline value, which is sort of the initial value. And it turns out that if you sort of do a first order Taylor series approximation, so first order Taylor series, you're going to end up with this expression here, that change in the bold signal is minus TE times delta R2 star. And since we're seeing a positive change, that must mean that delta R2 star is greater than 0. Okay? Or sort of less than 0. Sorry, less than 0. Right? So the change in the relaxation rate had to be negative. So the relaxation rate must have gone down. Okay. So now let's think about what causes relaxation rate. And this is going to get to we had the discussion in an earlier lecture about, what can cause like dropouts in human subjects. And so now we're going to dive a little deeper and look at the oxyhemoglobin molecule. Okay. So the idea is when those hemoglobin molecules go into your lungs, the oxygen binds to them. Okay, so that's shown in red here. Okay. And then as it goes into the tissue and the oxygen gets used up, then we start seeing these iron atoms. Okay, and there's 4 iron atoms for each molecule. So if I use up all my oxygen, I'm going to expose a lot of iron. If I use up some of the oxygen, I'll expose some of the iron. Okay. So basically, iron obviously is going to perturb the magnetic field around it. Okay. So in this case, for example, some of the oxygen is used up, so there's some field perturbation. I get some dephasing. If I use up a lot of oxygen, then there's going to be even more um, a dephasing, and therefore the signal is going to go up. Okay. So it's sort of interesting. We know that with activity, we said from activity, we go from here to here, like dark to light, right? So that means that we're actually, there's more deoxyhemoglobin, like more iron atoms exposed in the baseline state than when the brain's active. What you might have thought would be the other way around. The brain's active, so why isn't it using up more it's using up more oxygen, so why isn't there more iron there? right? So that was sort of the mystery. Like, why is the signal going up? We would expect it to go down. So the answer turned out to be something that was sort of figured out in the 90s, which is that there's two things, right? I'm using up oxygen, but I'm also can be delivering more blood, right? So there's two things. So one is I'm delivering oxygen through what's called cerebral blood flow. So if I can deliver more oxygen, I'm pumping up more blood, okay? And there's more oxygen that's available. And then I'm also I'm using up the oxygen through what's known as the cerebral metabolic rate of oxygen. And it turns out how much deoxyhemoglobin I have is dependent on the ratio of the metabolism divided by the flow. Okay, which makes sense. So the more I pump oxygen in there, that's going to tend to reduce the amount of deoxyhemoglobin, right? Because there's so much oxygen there. And so more CBF will tend to make deoxyhemoglobin go down. And then, but the more I use up oxygen, then I'm going to expose more iron and the deoxyhemoglobin will go up. All right. So there's that trade-off. So once again, increase in CBF makes the deoxyhemoglobin level go down, and an increase in metabolism makes that deoxyhemoglobin level go up. So those are the two competing factors. So we know because the MR signal went up, deoxyhemoglobin must have gone down. Okay. So therefore, we know that CBF must have won the game, right? There must have been more delivery than there was usage. Okay. And it turns out that that ratio is about 2 to 1. Okay. So for every increase in metabolism, the brain is going to deliver twice as much oxygen as needed. All right. And that sort of makes sense. Like if you're designing a system, like basically this system was designed, through thousands of years, millions of years of evolution, right? And so what it's saying is the people who survived, right? So everyone here and your ancestors, basically, um, it was advantageous to have more blood than you needed delivered to your brain, which sort of makes sense. You don't want to be like fighting a bear or your opponent and sort of run out of like cognitive power, right? Like, if the ratio is set at 1 and all of a sudden your brain starts getting slower or deprived of oxygen, you're probably not going to survive as well as someone who has plenty of oxygen available and can make better decisions than you. Okay. So that's sort of why it's exactly 2 to 1. We're not sure, but it sort of seems like, at least we know it's not 1 to 1. Okay. Yeah. Question. Yeah. I mean, the coupling ratio, at least um, certainly in other mammals that have been studied, it's sort of similar. Yeah. And I'm not sure. I don't know if people like, obviously we could probably find something like there's probably some sea dwelling. Like the problem is the brain structure changes so much over species. Like once you get into like, there's probably fish that live at 30,000 feet below and may not apply to them. But certainly for sort of any of your furry animals, it's going to apply. Yeah. Yeah. I mean, basically, I would probably venture to say that sort of, anyone, whatever I mean, obviously, so it's a good question. I don't know how well it applies to fish. I mean, sort of our evolution sort of came through like the fish line, right? In terms of somehow those became, things that turned into humans at some level. But yeah, anything close to us, yeah, it would be that ratio. So here's a nice graphic that one of my grad students made up for his PhD exam, which sort of gets the idea across. So essentially, we have cerebral blood flow that's introducing oxygen and hemoglobin. And then as it's being used up, then some of that gets converted into deoxyhemoglobin. Okay. And so typically what happens is your brain starts doing something, it starts using oxygen. So actually, if you look really closely in some studies, you actually there is evidence for this initial dip. Okay, especially if you can really a very active thing that basically the brain starts using up oxygen before more oxygen can be delivered, and there can be an increase in deoxyhemoglobin such that the signal goes down. So that's this initial dip. But very quickly, the brain's like, oh, I need more oxygen. So it very quickly delivers more than enough oxygen as at this 2-to-1 ratio. And that leads to this positive signal, right? So basically, you got a lot of oxyhemoglobin coming in, and so there's less deoxyhemoglobin. And then once things are done, then everything shuts off and it returns to baseline. There is this sort of post-stimulus dip that we're not really sure what that is yet, but I think um, the main point for this class is this positive bolt signal, which is sort of the basis of most fMRI analysis. Um, so so let's look at through these signals, some of these like one of these questions. So for example, an increase in the functional CBF response will tend to increase or decrease the bold signal. So we just said that, for example, if I have more blood delivered, I'm going to have more oxyhemoglobin. So that should tend to increase the bold signal, right? Because I have more oxyhemoglobin. Whereas if I have a decrease in the functional CMRO2 response, um, an increase in that response, then that's going to make much more oxygen being used up. So that will tend to decrease the bolt signal. So just by sort of understanding these factors, you can sort of see what's going on. So for example, here, the question is, in an FR experiment, the coupling ratio was initially set 2-to-1. And then what happens if the coupling ratio is 3-to-1? So that coupling ratio is how much does blood flow change with respect to metabolism change? Okay. And so the idea here is if it goes from 2-to-1 to 3-to-1, then from going from 2-to-1 to 3-to-1, I'm delivering a lot more blood at the 3-to-1 ratio. Therefore, I expect to see much bigger bolt signal. And so therefore, we would expect the magnitude of the positive bolt response to increase in that case. Okay. Okay, so now let's take a look at some of the applications. Now there's lots and lots of applications, so I'm just really going to give you a sort of a very quick overview of what we're looking at with fMRI. This is, I think most of you know this is Bart Simpson, right? Does everyone know The Simpsons, right? So this obviously is written in jest by the author of how Bart Simpson is using his brain. So most of it's for sleep and donuts and beer and other things. So interestingly enough, pretty much everything there has been studied with fMRI. So for example, there's been lots of fMRI studies of sleep, and it's actually being studied even more these days now that the relationship between sleep and like disease is becoming more evident. Lots of studies, including a lot here done on alcohol, where they look at, when you give people alcohol, what happens to their brains. Marketing is a huge area. There even used to be called this area, and might still be around, called neuromarketing, where they basically, if they can scam people while they're looking at different products, can they use that to figure out which products will sell better, right? Yeah. Um, Emotion, oh yeah, ads, right? TV time, right? They basically look at, how do you respond to commercials? Like, what's a better commercial? What's this one? Can't read what that says. What does it say there? Family, yeah, family time. Yeah, good. So basically, yeah, there's been a lot of study of, sort of how people remember things. Uh, Sex has been used a lot, has been studied. In fact, they've even put two people in a scanner at the same time while they're doing the activity just to see what's going on. I think the study was done in Italy or Europe or somewhere. I don't know. I don't think it was done in the States, but they actually had to build a special scanner that they had like could fit two heads in it at the same time while I think they were kissing. And I don't, someone can look up the study. I don't remember the actual details, but it was something that was a bit challenging to do, but they did do it. Um, let's see, what is this? Do's and don'ts, right? Okay. So, decision making has been a big sort of topic of study, even here, a lot of studies done at UCSD, where, for a long time, and I think it's still being done. I mean, especially with, issues of addiction and drug use, there's a question of like, why do people make decisions that are just not good for them, right? And so there was this thinking about, okay, is the decision making just different in people who have, say, addiction problems or not. Okay. There's even been studies, I don't think I put it here, of there was even some interesting studies done of, looking at, do people who vote Democratic versus Republican have different brains, and do they make decisions differently? And it turns out they do. Okay. So I think some of what we see is there's always going to be some amount of people on the right and some people on the left. And it's pretty much biology, right? People are just sort of, obviously upbringing has an effect, but there do seem to be some biological correlates to which way you will tend to vote. And then probably the people in the middle, will have a little bit of both. Okay. Um, I don't think there's not been a specific study I can find. If someone can find a study on donuts, let me know. That will be great. But there's been a lot of study of things like obesity and eating disorders. And so that's, in fact, here at UCSD, one of the main areas of study is like looking at eating disorders. Yeah.
Beginning of this, that they have the portable little MRI.
Yeah.
Sensitive.
Um, it's probably sensitive enough to do like really like if I flash a checkerboard at you, like to sort of see if you're alive or dead, you know. But there's probably easier ways of determining that. As far as I know, it hasn't been used. But if you find something, let me know. That I can add it to future years. Yeah. Yeah. And this is an old study, but it's still one of my favorite studies. So I'm going to present it to you. So this is sort of in the realm of marketing. And so here the study was basically um, of like wine tasting and how much do you like perceived wine. So for example, you're given wine in like a paper cup or something, and in some cases you're told that the wine costs $90. And then in another case, you're told that the wine costs $10. Okay. It's exactly the same wine, and you're asked to rate how much you like it, right? So this is the liking scale here. This is how much you like the wine. And so you can sort of see in this sample, if you're told it's $90, you really, really like the wine a lot. And the same wine, if you're told it's only $10, you're like, oh, this wine doesn't taste very good. Okay. Similarly affects a $45 wine versus $5 wine. Okay. So basically, price has a big impact on your liking of it. Okay. And then the liking without the price is almost the same. Right. So basically, you know, just being told that the wine's more expensive. And so what they did is they also did this in the scanner where they had people sipping the wine, and what you're seeing here is this is showing you areas in the brain that are responding to sort of that are looking at how your brain processes reward, okay, or pleasure. And so you can sort of see here. This is the $45 when you're told it's $45. There's sort of like, oh, I'm really getting a nice reward. I'm receiving pleasure. Whereas when it's $5, it's not quite as much. And in fact, there's almost like a negative response towards the end. So you sort of see this like negative dip. And same thing here with a $90 wine. You get this nice robust reward, like, oh, I'm getting a really nice reward. And here, just at $10, a little bump, and then just to like, this is, like you're disgusted with yourself for drinking $10 wine. Okay. So the key is that obviously there must be something in your brain that's computing how good this wine is. And clearly part of that computation is what you think the wine is worth. Okay. So just showing this way that people are looking at what are the actual biological correlates. And so this study can sort of show you what parts of the brains are trying to make that, what parts of your brain are involved in making that decision. Yeah. Yeah, the x-axis is time. Yeah. It's probably in seconds, right? So they're sipping the wine, and so this whole thing is occurring on the order of like 20 seconds.
They all have like a little bump at around 10 seconds. Yeah.
Let's see, is it seconds? We'd have to look up the study, but the bump where at 10 seconds?
Yeah, they all seem to have that little dip thing.
Yeah. I'm not sure that's statistically significant, and I don't really we'd have to look at the study again to see sort of how long they were sipping the wine for. So, yeah, but yeah, there, if you want to look it up, that the study, the thing is there. The other interesting thing that was sort of fashionable for a while, I'm not sure it's so fashionable these days, is sort of the question was, can I put someone in the scanner and figure out what they're thinking, right? Can I actually read their minds, right? And so this is probably becoming a little more practical now with AI and stuff, but and this work was done quite a while ago, and I'll just play the sound so you can hear it. Let's see.
The decoder built for
Right
primary.
here. See, I don't think you can hear that. So I'll just talk you through this. So basically, what you're seeing here on the left is the movie that was presented. Oops, what happened to my pen? Okay. So you're seeing this is what the subject is seeing. And then what they did was they put the person in the scanner and they made up of a lot of, they showed them a lot of different images. They almost basically, they just like the same way you train a deep learning network by showing a lot of images, they showed the subject lots and lots of images. And for every image, they looked at the brain response, right? So they know, okay, I have a pattern of brain response for every type of image, okay? And then they show them a new image and they say, okay, what collection of images added together would have given me that brain response? Okay, so it's sort of decoding that way. So this is an early attempt at trying to decode the movie. So you can sort of see here that it doesn't really tell you who it is. First of all, does anyone know who that actor is? Steve Martin. He's a famous comedian. He was, I think, believe this is from the Pink Panther series, which was a probably 70s or 80s. So it's so basically, at least it tells you, oh, there's a person there. Okay. So just by looking at the fMRI data, you can tell that the brain is perceiving a person. Okay. You can't tell who it is, but that's pretty good. I mean, and the person's actually in the right place too. Okay. Let's keep going.
Text on the upper left is the movie the subject
So now
saw.
it's showing like a dot, weird dots. So it sort of gets it. Okay.
Structural elements in movies like the.
So here you've got an elephant moving and, you know, it's sort of a blob. It's not doing a very good job, but it seems like there's something moving there.
You can see that this decoder does a fairly good job of recovering the structural information,
Yeah. I mean,
even though we're only decoding one brain area. This is a decoder that is recovering information from higher level visual areas that represent the semantic.
so here, like, I mean, this is actually doing much worse than a deep learning network, but you know, it's trying to label what the subject is seeing. So it does say, yeah, it looks like it's seeing a sky and a building and a body of water. So just sort of showing that there is information in the brain that it makes sense. We should be able to decode it. It's just how well we can do. Okay. So that's one area that's been going on.
So this is a decoder.
These are just some pictures showing the actual images here and then the decoded images down here. And you can sort of see, at least in this is from 2018, so it's quite old now, but you can sort of see it's doing a reasonable job of at least getting the broad outlines. I'm not sure there's any project on this, but certainly it'd be interesting to see what the latest is on this. And then the other thing is sort of relates to this quote that Plato made many, thousands of years ago that reality is created by the mind, and we can change our reality by changing our mind. And so one of the ideas is, can you actually change your mind? Can you actually control your mind? And so this was an area that is probably not in vogue nowadays so much, but at least it's an interesting area, which was called real-time fMRI, was can you actually use the fMRI signal to help people train how to control their minds? Okay. So in this case, for example, what they did was they looked at this area of the brain and this area of the brain, right? And essentially they showed subjects like the signal in that area of the brain, and then during certain periods they said, okay, make the signal go down, and then it turned out that the subjects with enough training could actually control the signal in that part of their brain just with feedback. Okay. So they don't know really how they're doing it, but they're just doing it. So imagine without feedback, I told all of you to increase or decrease the fMRI signal in your posterior cingulate cortex, right? Are you guys doing that? Okay. Okay. Now increase the signal in your hippocampus. Okay. Decrease the signal in your amygdala. So it's very hard to do that, right? First of all, you don't really know where it is, but it turns out that just like anything else, just like kids can learn how to walk or bike, if I give you the right feedback, you can figure out how to do that. So this was being used early on. The hope was that people could use that to, for example, treat things like addiction, like you could maybe have people sort of reduce areas of the brain that are related to addiction or maybe even depression. But I think the results, it's just too difficult of a technology so that I don't think at this point it's been successful, but at least it's a proof of concept that people can modulate their brains. But this is a very expensive form of neurofeedback. And then finally, we'll end today with talking a little bit about what's the difference between what's called task-related bold and resting state fMRI. And so pretty much everything we've talked about so far is you're telling the brain what to do. You're saying, okay, do a task, look at some light or make a decision, and then don't do it. And then you're looking for that contrast. But it turns out if I look at a brain over time, I look at the bold signal, it's constantly changing. And right now, if I were to put each of you in an fMRI machine, I would see images like this. Things are just like moving all over the place all the time. So it looks like a lot of noise, right? In fact, if I looked at any given part of your brain, this might be the fMRI signal I would see. So it looks really, really noisy, right? And so for many years, it was just thought, okay, well, there's just a lot of noise in the brain. But then it turned out that here's an interesting thing. If I ask you to tap your fingers, right, like that, and I ask what areas, and I have you tap like, for 20 seconds and don't tap, and then tap for another 20 seconds and don't tap. And I ask what area of the brains have the same up-down activation, then I would map out those areas of the brain. And these are your motor cortices. This is the part of your brain that controls your motor function. Okay. But now here's the other thing. I can just have you rest in the scanner, not do anything. And if I look at a signal, let's say the green signal in the left side of your motor cortex, and I look at the signal on the right side, it turns out even when you're not doing anything, those signals are very correlated, right? So then that means I can just take a signal from, let's say, one area of the brain and ask what other parts of the brain are changing like that part of the brain, have signals that look similar, are very highly correlated. And then it turns out that the map I get is almost the same as I would get if they were doing a task. So it turns out that the brain is always talking to itself. It's always sort of somehow connected to itself and sort of having correlated activity. Okay. So this has led to this huge area called resting state fMRI. Yep.
In some cases, there seems to be a bit of like lag between two curves. Is that something special, or it's just...
You mean the green and the blue?
Yeah, yeah.
Yeah, they're not going to be perfectly correlated. First of all, there's noisy, right? There's a lot of noise either due to the acquisition or just there's just lots of sources of noise, right? So it's not going to be perfectly correlated. Yeah. Like even two areas of the brain. I mean, also the brain, like we don't really understand the brain that well, but each brain area, how it's working with another brain area, there's going to be very complicated dynamics. So even if we had no noise, we wouldn't expect them to be perfectly correlated because your left and right sides aren't doing exactly the same thing, right? They're not doing the same control, right? So, for example, even if you're just like, clearly the thing is, even if you're just lying there in the scanner doing nothing, it's not like your brain is turned off. Maybe you're thinking about lunch, maybe you're thinking about, driving. And so even there, like if you're thinking about driving, that might activate one part of your brain more than the other. Yeah. So it's resting state, but it's more like lying in the scanner trying to do nothing. Okay. Yeah.
They're like scanning, meditating, because like you're supposed to kind of turn your brain off.
Yeah, we've done quite a few of those studies. Even when your brain is, there's a whole area. I don't have slides on them though. There is areas and there does tend to be we'll get to this. Let me get to this thing and I'll show you what address your question. Okay. So an interesting thing about this is, for example, one of the things that we found that reduces connectivity is if you give the person caffeine. And so, for example, this is the resting state connectivity before caffeine and then after caffeine. in a lot of subjects, it just gets rid of the connectivity. Okay. And then to get your question, there's been, now like hundreds of networks discovered by just doing resting state, but the most ones that are sort of always seen are, clearly there's one related to your visual network and your motor network, but probably one of the most famous ones, which gets to your question, is what's called the default mode network. Okay, let me see if I have a slide on that. And I don't. Okay. So the default mode network is active when you're thinking about yourself. Okay. And when, like, so let's say you're just like sitting there thinking about lunch or thinking about, school or whatever, or, your friend or your parents or your family, whenever you're thinking about yourself, that network is really active, right? And then when you have to do a task, like you have to go shopping or you have to like, make a phone call or do something, then that network tends to go down. Okay. And then another network called the attention network becomes more strong. And so it turns out those networks sort of go in and out. Like the more you're using the one, the less the other one is active. So one of the things that people have found is with meditation, you can sort of reduce the activity in the default mode network, right? Which essentially you're reducing your sense of being obsessed with yourself. Okay. And actually, in fact, people can use like real-time fMRI to train themselves to reduce their activity in the default mode network. And actually, it turns out that experienced meditators can do that much better than normal people. Okay. So basically they can, because a lot of reason why people do meditation is because they're suffering or, something isn't right. And so a lot of that is just reducing this, because if you have less sense of self, then, what problems are there in the world, right? It's not like you having the problem. There's just things going on. Okay. So yeah, there is a lot of stuff done on meditation. Yeah. And it turns out that we can even do something called fingerprinting. So basically, I think this was a study of 100 subjects, and this was from a former grad student. We just looked at the resting state networks from just like 30 seconds of your resting state data. And the question was, out of 100 subjects, could we predict, like if I scanned you on one day and brought everyone back the second day, can I predict who's who just by looking at your resting state network? And it turns out with like 99.5% accuracy, each of us has this signature. So basically, the way your brain is talking to itself is actually unique to you. It's like your fingerprint. Okay. So there is a lot of information available in what your brain is doing. Okay, so I think what we'll do is we'll end here for today. And then we'll pick up on Monday with talking about sort of the remainder of our sort of lectures will be more focused on. We'll spend some time looking at diffusion and then flow and then other sort of future sort of what's the future of fMRI and MRI in general. Okay. So I think the only things logistically are, yeah, I mean, if you have questions about your project, please ask about them. Otherwise, I think homework 5, I guess we'll extend the deadline. Is that right? So we'll extend the deadline for homework 5 till Saturday, just because I guess some people need a little more time. And then we'll make the optional extra credit problems available like Friday or something. So if people want to do those, but those are just for extra credit. Okay. With that, if there's no more questions, we'll end for today and answer questions after. Okay, thanks.